Thank you very much for coming. So this is not going to be a talk about cancer as a clinical condition. And cancer means a lot of different things to a lot of different people, families with cancer or people with cancer. Cancer as a doctor, as a nurse. I'm not going to be talking about that, and I'm not qualified to. My son, incidentally, is an oncologist at Guy's Hospital, and he can do that. And he's got that bedside manner, which I'm a scientist, you know. We don't have bedside manner. What's it mean? So I'm going to tell you a little bit about the biology of cancer and try, if I can, to convey to you that it's something that's not evil or fearful. It's a natural part of biology and a part of biology that's gone wrong. There's wonky software, and we are in the process of fixing it. So the story is all good. So I'm going to divide the talk into 4 parts. First of all, what is cancer? Second, why do we get it? Why is it so difficult to treat? And then what are people like me doing about it? And as you know, cancer is supported by a lot of charitable groups and organizations. And it is responsible for the second biggest killer in the world, at least in what we fondly call the developing world and developed world. So what is cancer? Well, cancer can be really defined here as the inappropriate and unregulated expansion of somatic cells, potentially leading to death. Now, some of you won't be familiar with what some of those words mean, but there are important words here that give you a very good idea about why cancer is such a weird thing. First of all, there's the inappropriate. So that if you think about something being inappropriate, you've got to define what's appropriate. So how does the body know when a tissue is appropriate and when it's inappropriate? We'll come on to that. Unregulated, so that means that we're dealing with something that normally is very tightly regulated, that loses its regulation. We're not dealing with new things, new alien beasts or anything else like that. We're dealing with common or garden cellular and body processes, but ones that somehow have got out of hand and running rogue. Expansion, that reminds us that cells are born and they die. And it's the balance between the two that matters. So in cancer, you get relentless expansion of cells, but you also get a huge amount of cell death and cell suicide. But the cell growth and expansion exceeds that of the cell death. Somatic cells, well, we've got 2 types of cell in our body. Somatic cells, body cells, and germ cells. So this is the disease of our body cells, and there's lots of them, as we'll come on to shortly. And then the question is, why does it lead to death? And that's really a very difficult question to answer. Different cancers cause different side effects and pathologies. And so it's complicated why they actually are deadly, but they're left unattended. But nowadays we have ways to fix that. So I've shown you here the key words that are important here. And we're going to go through them and encounter them in this talk. So first of all, cancer is a common disease. I think we all accept that. The lifetime risk of cancer is around about 50%, and up to half may die of their disease, so that's declining in the last 20 years quite rapidly. So cancer is a common disease, but we have to remember that cancers are clonal. That is, they arise from a single cell within the body, and that aberrant cell then proliferates to form a tumor. Now, how many cells do we have in our body? The answer is a large number. So each human contains about 10 to the 14. That's 1 with 14 zeros after it. 100 trillion, I think, is the right way to express that. So to give you some idea, there's 100 billion stars in the Milky Way. That's a huge number. But we've got more cells than 10 Milky Ways, more cells than 100 Milky Ways. We've got about the same number of cells as 100 Milky Ways. So it's a huge number of cells. And in principle, each one of those cells has the capacity to turn into a cancer cell if it gets damaged or corrupted in some particular way. So actually, the cancer is amazingly rare. The cancer cell is amazingly rare. So what we've got here is a conundrum, which is cancer is both very common and very rare. It's common because lots of people get it. But it's rare because it arises very, very rarely within the huge number of cells within each individual. So, who gets cancer? Well, the answer is all multicellular organisms get cancer. Single-celled organisms can't get cancer. And the reason they can't get cancer is they sort of are a cancer. They're just cells wandering around eating and gobbling and going mad. But this shows you, for example, that cancers arise in plants. There's one of the tree. In dogs, that's not the dog by the tree, by the way, just so to reassure you. The dog. This is a fruit fly with an ectopic growth growing out of its eyes and its head due to a mutation in a gene that drives replication of cells. That's a carp of some kind. I'm not very good on fish, I'm sorry, but anyway. And yes, even sharks get cancer. So all multicellular organisms are prone to cancer. And the best way to think about this in general is the cells in our body have got software that tells them what to do and programs that they run in some way, in biological way. And those cells, every one of those cells is at risk of turning into a cancer cell during your lifetime or somebody's lifetime. So how and why do we get cancer? Well, the reason for this is interesting and fascinating, and really where the edge of cancer biology is at the moment. Essentially, cancers are there because we've got machinery for cancer embedded in every one of our cells. So this isn't something gone completely crazy. This is a program that has got corrupted, but a normal program. Now, when building and repairing normal tissues, they regenerate very rapidly. If you cut your finger, there's a lot of swearing and moaning and everything else. You put a Band-Aid on it, it bleeds for a bit, then it clots. And then it goes through a process of healing. And during this process of healing, lots of things happen. First of all, there's injury, inflammation. There's migration of the cells around the injury, migrating towards each other to seal the wound. And then there's resolution and polishing it off at the end, which sometimes, more often than not, leaves a little scar, but nonetheless makes sure that that tissue is whole again and can work. And the whole reason this has evolved is if you're bitten by a saber-toothed tiger and you survive, you've got to get up on your feet as quickly as possible and run away. So this wounding process is as quick as it can be, but it's more like a Band-Aid, a sticking plaster. It's not an accurate refixing of things, but one that will do well enough. And well enough is good in evolution, as we'll see a bit later. So normal tissues do what cancers do. The cells proliferate, they become more plastic, that is, they change their identity. If you think about it, a cell that's going to come into a wound to try and fix it, an inflammatory cell which comes in to clean up bacteria or anything else in the wound, it doesn't quite know what it's going to face, so it has to basically improvise when it gets there and turn into lots of different subcells doing different things. So to do that, these cells retreat from their normal function and they become more plastic and oozy and everything else. And that happens in normal injury, and cancer cells show that plasticity. They show massive amounts of that plasticity. And that's one of the reasons that they're difficult to treat, because they wander and they evade our ways of treating them. And we'll come on to that later. Cells in an injury, just like cancer cells, migrate and spread. They recruit other cell types to drive inflammation, suppress local immunity, and drive the formation of blood vessels, all of which are needed to mend the tissue that has become damaged. And here's a picture of what it looks like. On the left, you see shortly after somebody's cut their finger with a bread knife, which I did only last week. It made all the noises that people make. We see a clot of blood, and then lots of cells coming, roaring in from the bloodstream, and from the bone marrow where these cells are born, these blood cells, and they're coming in and they're cleaning up the whole business. And that turns into, after a few days, to a granuloma here in the middle. And if we look at a tumor's basically this middle phase stuck on and just overwhelming the tissue around it. Because there's no injury, this is being driven by a mutation in the cancer, in the tumor cell. So there are huge similarities, the same programs are running, much the same programs are running, but to a first degree you can say that the normal tissue knows when to stop and the cancer cell can't stop or doesn't know when to stop. In fact, cells don't know anything, they're robots, so it can't stop for some reason. It's got trapped, stuck in the go position. And that tells us that what we already know, which is that cells that make up our body basically are robots. They do what they're told to. They take information from outside, and I've depicted this as a sort of microprocessor connected to a TV set or whatever. They take information from outside, neighboring cells from the body, Things like, whether you've got a temperature, whether you're overweight, whether you've got some other disease or pathology that makes you inflamed or something else like that, or whether you're feeling fit and healthy and just come back from a run and grazed your knee. They take all this information together and they process it and they run different programs depending on which type of cell they are. Now, there's some flexibility between these programs, so some that work Microsoft can also work Photoshop, others can't. But by and large, they run a set of programs that are related and that have evolved to deal with these little robots, these cell robots, and keeping them in order. But these cells don't do anything. They don't proliferate, they don't differentiate, they don't even live unless they get single signals telling them all the time to do so. So they're slaves. And the machinery, this Intel Pentium is not, well, we don't have an Intel Pentium in our cells. Please accept that. But this is what it looks like, a very greatly simplified map of all the different biochemical reactions that are going on, which run our individual cells, albeit specialized in different ways for different types of cells. Skin cells are different from stomach cells, they're different from lung cells, and so on and so forth. Now, these cells are very tiny, but they've got a lot of information in them. So this is just what happens when you empty the vacuum cleaner. Watch all the dust you pick up. I like these photos because they make me go, really? I did that? I cleaned that up? So these are dead cells, but you can see their general size and shape. Skin cells that are being found in a vacuum cleaner bag. And that's what they look like, and that's what most of the dust that you hoover up when you clean the house. If you clean the house. And that's a creature who lives off this. We all have them. Sorry. Okay, there's a little dust mite there, and they think this is wonderful food. This is like going to the supermarket for them. So how much information do we have? Well, each cell in our body has about 2 meters, 2 yards of DNA. If you stretched all the DNA out in a single cell, it would be further than I could reach out here. And that means in those 100,000 billion cells, there's enough DNA, if you stretched it all or connected it all up and aligned it and ran it all together, to go to the planet Neptune and back 10 times. That's in your body. And these 2 meters of DNA are wrapped up and packaged and packaged and packaged and packaged in a thing so small you can't even see it. So there's a lot of information, a lot of stuff going on, a lot of room for errors to accumulate when DNA is copied. Because when DNA is replicated, which is the genetic material of the cell, it needs to be copied one time, and the copies are always slightly different to the originals because of errors that creep in. And these errors gradually accumulate over time. And as they accumulate over time, they give rise to clonal progeny, baby cells, and a baby cell with a mutation divides, and its 2 daughter cells have those mutations as well, and so on and so forth. And so this is the process of how cancers arise and how they so-called progress, which is they basically become, the point is you don't see the cancers that don't progress, you only see the cancers that do progress. So the great paradox of cancer then turns out to be this, which is that high and rapid rates of cell proliferation are necessary in many adult tissues to fix things that go wrong. Some of your tissues, many of your tissues like the intestine, like your skin, like your bone marrow are replicating all of the time and renewing. But many tissues will renew only if they're damaged or infected or so on and so forth. So many adult tissues are dividing and proliferating either continuously or occasionally. But this proliferation has to be in the right place at the right time, and it has to be very easy to activate. So when you cut your finger, you don't want to sort of debate in your brain and your parliament of all your cell type meeting once every month and going, yeah, well, he cut his finger last week. Who's in favor of healing it? Okay. No, it's got to be bang, immediate. So this has got to be a very easy program to drive and activate. At the same time, cancers have to be very rare, so this can't go wrong. So this has to be very difficult to activate these programs in the wrong place at the wrong time. So essentially, you're asking for something to be incredibly easy and incredibly difficult at the same time. So Am I mad? Can you do that? The answer is no, we all do that every day. Well, most of us do it every day. We have a thing called a key. And what does a key do? The key turns a combination. So it turns out that combinations of signals are what give the cancer cell and the normal cell their specificity. And in order to get a cell to replicate, you have to have several switches flick at the same time. Let me give you an example here. Growing cells minimally need two types of signal. They need a signal to grow, and they need a signal to survive. And these two signals are connected. What do I mean by that? Well, what I mean by that is that the machinery that drives cell growth is also a death machinery, a suicide machinery. So every cell that picks up the machinery to proliferate and do this dangerous thing, which cancer cells get stuck doing, every cell that does that also picks up a suicide bomb and will probably die unless it's rescued by a signal from another cell keeping it alive. So all of our cells are interdependent on each other for their very survival. So the cell dies if the growth machinery is activated without that survival signal. But correspondingly, when the cell next door provides a survival signal, it sends out a signal that suppresses the death in the yellow, but these signals also suppress the growth of the cell. So either one of these, this stops the cell. So either one of these processes leads to no cell replication because the growth machinery kills the cell and the survival machinery arrests the cell and stops it from dividing. But now you put the two together and something really magical happens because the growth machinery still runs, but the death machinery is now being suppressed. So what you get is growth and survival. So only when these two things interlock together do you actually get the right signal. And so what we're talking about here is essentially a combination lock. We all use them all the time. We make things easy because we have a combination, bumps on a key, safe locks like this, and so on and so forth. So this isn't something that we've invented as human beings. This is something that's built and hardwired into biology. So that's why cancer is so rare. It's because signals to do the things that cancer cells do are normally very, very tightly controlled. And one way of thinking about it would be, it's a bit like putting your foot on the accelerator pedal in a car. Now, what happens when you take your foot off the accelerator pedal? Well, hopefully, the car slows down. It's rigged so that it defaults, that it's in basal state, is to stop. So when you put and these analogies don't work in modern cars because you've got cruise control, but just ignore that. Okay, it's stuck in the 1990s. So essentially what happens is that you put your foot on the accelerator pedal, the car moves forward, you take your foot off the accelerator pedal and the car slows and stops. So the default state is to stop, the safe place is to stop, and only when being actively told to divide and kept alive at the same time do you actually get cell growth. So this is a very tightly controlled and very tightly constrained phenomenon. All right, so why is cancer so difficult to treat? And here I want to talk a little bit about what I might call the mythology of cancer. So first of all, it's cancer cells are unkillable. Second, they're mean, malevolent, cunning, planning, conniving, hiding, strategizing. They're fighting a battle with you and with the patient. And third, they're so complicated and complex that you're a bit like going in to fix a laptop with a screwdriver by just banging. Well, in fact, I've done that, but still, never mind. By banging the screwdriver into the motherboard. So let's take a look at these. Are cancer cells really unkillable? The answer is, of course not. Here's 3 examples of something that kill cancer cells. Sulfuric acid, nitric acid, and hydrochloric acid, particularly the concentrated kind. You pour that on a dish of cancer cells, they're dead. Okay? It's 100% effective, no reported resistance. Well, of course, this tells us that the problem is not that cancer cells are impossible to kill, it's that they're very difficult to kill inside the body of the patient because the normal cells are also sensitive to many of the things that cancer cells will kill. And in many ways, the search until relatively recently for cancer therapies has been about trying to find things that kill the cancer without killing the patient or damaging the patient's tissues too much. And of course, what we want is we want to refine that, and we are in the process of refining that more and more and more as we understand what the differences are between normal cells and cancer cells. So what about the fact that cancers are in a battle with the patient? They're malevolent, cunning, planning, conniving, hiding, strategizing. Well, here we've got to turn to a little bit of understanding based upon evolutionary biology. So, are they really cunning? They look like they are. But the truth is that any process that results in heritable changes, that is, changes where one being, one entity divides in two and then gives its information to the two daughter entities, anything that's heritable in a population, some change or anything else, will spread over many generations if it confers a selective advantage. That is, it's quite good to primitive humans would have run sort of more or less on their hind legs, but doing a lot on all fours. Standing up and losing hair was really great in the African plain, because that meant that human beings could run faster and for longer because they could sweat. Dogs, for example, don't sweat very well. They pant and they sweat through their pores, but they don't sweat. And they're very, very good at running quickly, or at least my dog is, running quickly for short distances. Then they cop out and say, give the ball to someone else. Okay. But we can run long distances because we sweat and keep our temperatures down. So that's an example of a trait that once built into the human body got accelerated and distributed around very quickly amongst all humans because they had survival advantages. So this is what we're dealing with. Evolution is fueled by variation. So populations have to vary, and they do vary because nobody's the same as everybody else, and everyone's got different genes and different versions of different genes. There are random differences between individuals within a population. That comes under selection, which is some of these random variations give more of an advantage to others than others, and favor some of the population over the rest. And it appears that evolution is moving in a particular direction, but actually evolution has no direction. It just seems that way because we only see the winners. We don't see the quadrillions of losers. And I'll give you an example of that now. So evolution is usually depicted as progression, which we start out with hairy ones and we move to slightly less hairy ones at the other end. And in fact, this is a process that is continuing, but of course it doesn't always go in the right direction, by which we mean more taller, greater athletic humans than before. It's sort of going in this direction now, which is... and in fact, sensible people would say it should really go in this direction, which is... go back, we mucked up everything, you know. So if human beings do end up destroying the world, it will be a failed evolutionary experiment. So let's go back 66 million years when dinosaurs, we all love dinosaurs, okay. Dinosaurs were ruling the earth, and mammals were sort of hiding in little holes everywhere and being very small like shrews. Well, we know that they died out 66 million years ago, and the reason they died out was because an asteroid hit the earth in the Yucatan Peninsula. Now, dinosaurs, it was pretty bad. It's pretty bad for mammals too, but not so bad that it wiped mammals out. It wiped out the dinosaurs. So people sometimes say, thank God for the asteroid, otherwise we'd be sitting here with our little forelimbs going, okay. Horrible idea, sorry. So the thing that really reshaped the world was the plankton in the oceans. So the plankton in the oceans at this time was full of very diverse plankton, the tiny little plants and plant-like single cells in the ocean. Coccoliths, dinoflagellates, radiolarians, and these were hugging the warm equatorial waters where it's always sunny. They would photosynthesize over green, and they would be living there. And these poor diatoms were just on the way out. They were exiled to the poles where it's cold and dark half the year. They were really on the way out. So if you look at that, you say, well, it's pretty obvious that the diatoms are on the way out and the coccoliths, the dinoflagellates and radiolarians are going to win. Then along comes the asteroid and bush, off it goes. Throws up loads of dust and muck and everything else like that, and everything goes dark on the Earth for probably a year or two. Well, during that year, there's no photosynthesis. So guess what? Who's already pre-adapted to living in the dark for 6 months at a time? Yes, that's right, the diatoms. So these guys, the coccoliths, dinoflagellates and radiolarians, no sun, no photosynthesis, they all die off. And guess who wins? The losers are the winners. So this is how evolution works. It's not a progressive process. It's luck, randomness, all sorts of things. And we don't see the losers. And the same is true of cancers. We don't see the cancer cells that are so corrupted that they don't work anymore and they fall apart and they die. We only see the ones that can get through that. And it gives us the impression that cancers are progressing, but they're not progressing. They're just moving around in evolutionary space. So go figure. So this sense of evolution starting with simple and going to more complex and then things dying out and everything else has no directionality built into it, apart from the fact that obviously when life started, it had to be as simple as possible, because that was the beginning. And it's got more complex, but not in any particular way. And there are just as many organisms which are basically going in the direction of becoming parasites and losing their limbs and eyes and everything else, as there are organisms gaining eyes and limbs and various other sort of things. It's not got a built-in direction. And the reason that can be misleading is this, and you probably know this, is so during the Second World War, there was a ministry committee of engineers put together to try and work out how to improve the survival of airplane pilots. And so what they did was they noted where all the bullet holes were. And most of the bullet holes were in the wings and the mid-fuselage. So they thought, well, this must be, it's a large area, this must be where the bullets go. That was completely wrong. It turns out that the bullets that hit the cockpit never came back. So these are the survivors. And that's the way we have to look at evolution. You have to think again when you look at how things change evolutionarily. And as a cancer biologist, you also have to look at how things change and what they mean. So evolution looks like it has a plan, it looks like it's got a direction, it looks like it has a purpose, but actually it has none of these. So cancers are not malevolent and cunning and everything else. They're just evolving in evolutionary space and unraveling and diversifying and everything else. And we don't see the losers, we only see the winners, which are the losers for the patients. And then the last reason is that cancers are very complex. Biology is somehow, too complex for us to really understand. That may be true, but I think it's unlikely. I think we can do it. We're just working our way through it at the moment. This, for example, is the mutational map on the left of pancreatic cancers. So what you can see is each type of mutation has a different color, that these tumors, these cancers in patients accumulate lots and lots of mutations. And on the other side, we have human lung adenocarcinoma. The same is true for that. So you need so many mutations to make a cancer, and they're different from each cancer patient to each cancer patient. In fact, each cell within a cancer is probably different from every other cell within a cancer. How on earth are we going to have to be able to understand what's going on, and then, having understood it, fix it? Well, complexity can be misleading. So here are granulomas. Granulomas were known to be disease markers in the 19th century of a disease called consumption. So they would form in lungs and then people would die. It's tuberculosis. And people didn't know why they looked different. They came in various shapes and sizes. They were given names, put in boxes. Arguments were made about what they really do and what the different cells in each of these granulomas really do. But it turns out that the most important feature is that every one of them is induced by a single bacterial type. So the Mycobacteria tuberculosis. So it turns out that how these things look when they present themselves is actually less important than what makes them look that way. And if you treat all of those granulomas with the right antibiotics, they hit this, knock these bugs out, and the granulomas go away and the disease retreats and the patient is cured. So we don't necessarily have to know all the intricacies of cancer in order to understand how to treat them. It's just the same. So cancers, I don't believe, are too complicated and too complex to unravel. And it turns out there's more in common across different cancers than there is differences. So there are lots of differences. Humans love differences. They make patterns and they can present them as those charts with colored things. They look really good. But the point is, if we go back to that slide, are there ones that always occur? And at the top, you'll see the ones in blue of both of these, and in the pancreas and in orange and greeny type things. These are the common mutations, and these are probably the mission critical mutations that drive this disease, upon which all the other mutations reside. So not everything, for example, If I told you tigers are dangerous and they have stripes, you wouldn't say, oh, the most dangerous thing about the tiger is its stripes. I must avoid striped animals. I've got a cat, it's striped. It's also the same order of organism. It's a feline, but it's not dangerous, though sometimes it can be. So we look in here, we can look to see the diversity, or we can see the commonality. Again, there are people in the Crick and many other organizations around the world trying to look at both of those features. Not everything about cancer is immeasurably complex. Okay, so where are we? What can we do about it? What can we do about cancer? Well, here's our map, if you remember this simplified map of the microprocessor that is the cell. Greatly simplified, it has to be said. And no one really can put in their brain, because this is also a static picture. Everything's moving around and gurgling. Thank God it doesn't make too much noise. Can you imagine if your cells gurgled all the time? So where do we go? Well, we know now pretty much all of the pathways that get corrupted in cancer. And by now, after 20 years of developing new cancer drugs, we're able to target almost all of them. So you think, well, why is cancer not treatable or curable yet every time? The answer is it is treatable and it is curable in some of the times, but that's because there's lots of redundancy in the signaling pathways here. By redundancy, I mean there's one way, if you block this pathway here, then another one will take over and so on and so forth. And that's because biological systems that have to work all the time have to be resilient. So to be resilient, they tend to build up redundancy within the system, so there's lots of different ways to cook an egg, for example. So, but nonetheless, we've got lots and lots of drugs. So why aren't these drugs working all the time? Well, the problem really we've got at the moment is all we can do is hit them with a screwdriver or a hammer and knock them out. We can inhibit them. And if, for example, your accelerator pedal in the car gets stuck down, what you'd really like to do is fix the spring that moves it back up again. But instead, all we've really got is things that like sledgehammers to smash it to pieces and break it. That's the first stage of developing new therapies. The new stage of developing therapies is to put right what's actually gone wrong in these molecular switches, rather than just block them. And that's a very exciting area that we're involved in around the world at the moment. And there's also good news, because we know we have very potent cancer protection devices within our own bodies and our own cells. And we know this for a very simple reason. Here's an elephant, largest land mammal, largest land animal ever. No, probably since the dinosaurs, but we killed those off, didn't we? Yeah, that's right. There's an elephant shrew. The elephant shrew is about 1 million times smaller than the African elephant. And so it has a sort of 1 million times less cells. So let's put them in perspective. There we are. Okay. 1 million times less cells. Now, if cancer cells just have a given probability of turning into a cancer cell, if cells in the body have a probability of turning into a cancer cell, then that probability should be the same for both of these organisms. And because you've got 1 million times more cells in an elephant, they should get 1 million times more cancer. It's 1 million times more chance. And that's an elephant, but whoa. And on it goes. So that means that a blue whale can live for 130 years, be, 100 times the size of a human being, and not get any more cancers than a human being. That is 1 in 2 blue whales. I don't know the frequency of cancers in blue whales. They're a very difficult experimental model to work with. Okay, but we know from observations at humpback whales, at least, that humpback whales have much reduced risk of developing cancer from each of their individual cells than we do, and we have much more protection than the elephant shrew does. So it's not a zero-sum game. It's not like we can't thwart our evolutionary legacy for cancer protection. We can make it at least 10,000 times better, in which case cancer would cease to be a public health issue whatsoever. And the other thing that we know is that injuries that I told you about before normally do heal by themselves. So if you've got a cancer which is stuck in that mode, there's a program in your cells and in your bodies to shut that program down. We just don't yet know how to access that program. But we've all got it because we've got it and you can see it working every time you cut your finger and it heals itself. It's an anti-tumor mechanism. And then I think the most amazing weapon that we've got is that we've got these enhanced, we can now enhance and assist those innate protective mechanisms. We're all actively protected against cancer all of the time to varying degrees. We're probably not protected as much as possible. In fact, we know we're not protected as much as possible. And the reason for that is that we probably don't need to be protected. We need to basically move our genes around and then evolution's done its work. So all of that going to parties in your 60s and 70s and everything else is just bonus time. But we can fix it. We can fix it. And here's what we're doing. We've got a vaccine against papillomavirus. We've got targeted drugs against breast cancer. The list is huge now. And each of these different drugs, this is a vaccine, of course, and this is a therapeutic drug. Each of these different modalities are being developed again and again and again and improved all of the time. And the picture is very rosy. For example, we've rendered human papillomavirus and cervical cancer almost wiped it out amongst humans now through the immunization strategy with Gardasil. So we've got targeted therapies. We can exploit our potent natural differences against cancers. But the most important device we've got is human endeavor and creativity. And cancer research isn't just something that cancer researchers do. It's something that entire nations do and communities do. And you're all part of that community. So what we've got is we've got courage and trust. We have to have the courage to do the difficult experiments and to think outside the box. And you have to please trust us to do it and do it to the best of our ability. It's our life's work. So that's us at the Crick. This is a great place to welcome you. Thank you for coming and thank you for listening.